Hello, everyone. Welcome to week number 3 in Bio 120. This is a good week, one of my favorite weeks, because we're talking about bacteria and antibiotic resistance, and you're starting your final project. So this is a great week. Let's see what we've got going on. First of all, a quick review of last week. We looked at macromolecules, which included carbohydrates, proteins, lipids, and nucleic acids, and the functions of those groups of macromolecules that make up organisms. We learned about the importance of water in cells, and we talked about carbon as the basic element of life. And why? That's because it creates complex and very large molecules that we call macromolecules. So we studied about the chemistry of life last week, and we will use all of those ideas through the coming weeks. This week is all about classification of organisms, the types of organisms, and their cell structures. So this week is about cells. So why is classification important? Because when we group organisms into classes, then that determines that these particular organisms have all the same characteristics that we explain, okay? The basic groups of organisms in the world are grouped into domains. And so there are 3 domains. Eubacteria, which means true bacteria. Archaea, which contain the archaebacteria. We used to consider them ancient. We don't now, but they're very different. And then eukarya, which contains all of the eukaryotes. So let's talk about those 3 things, those 3 types of organisms for just a second. The eubacteria are found everywhere that you normally think of in life. So on your body, in the soil, in the leftover food that's incubating in your refrigerator, everywhere in your water sources. Some of them float around in the air that we breathe, just everywhere. Those are the true bacteria in the domain eubacteria. Then I also explained that there's another domain that contains bacteria, and those are the archaebacteria in the domain archaea. Archaebacteria live in the most extreme environments on the planet, down in the very depths of the ocean, in hot springs where nothing else can grow, in the saltiest bodies of water on the planet, at tremendous amounts of pressure, and that would be under the sea. In ice, we find archaebacteria that live in extreme environments. These we don't usually come in contact with in life. And then we have the domain eukarya, and that contains eukaryotes. Well, think about what I've talked about so far. 2 groups of organisms that are bacteria, and so that means every other category of life form is in the domain eukarya, and that is true. All the rest of us are eukaryotes. So who does that include? All of the animals, all of the plants, all of the fungi, all of the protists. Protists include algae and amoeba, lots of different strange little organisms that are grouped in the category of protists. So all the rest of us are eukaryotes. So we got prokaryotes, which are any of the bacteria, and we got eukaryotes, which we're in that category, right? Eu means true. Kary means kernel, which refers to a nucleus. So prokaryote means before nucleus, and eukaryote means that we have a true nucleus. This tells you something about our cell structures. Bacteria cell structures are totally different from ours. Now, we have a lot of molecules the same, but our cell structures are very different. Bacterial cell structures are very simple. They're not little compartments inside bacterial cells. They aren't big enough to have compartments in them. So the water that's floating around inside the cell is in contact with the DNA that's floating around in the cell, which is in contact with all the enzymes that are there, the proteins that are made, the carbohydrates, etc., etc., etc. Eukaryotic cells, on the other hand, have all of these little compartments, like a nucleus, like mitochondria, like the Golgi apparatus. Oh, there's so many little compartments or organelles that eukaryotic cells have. So we have very different cell structures. This is going to be important when we get to our discussion board postings this week. Okay, so that's a little bit of the biology. The chapters in the OpenStax textbook. Read Chapter 20, Section 1. This will help you this week. Go through Chapter 4. You can skim it. That's all right, because it's all about cells. And you need to know the cell structures in order to answer your questions this week. But definitely read Chapter 20, Section 1. That will help you. Then read the Module 3 overview and watch the videos. The Invisible Universe is just wonderful. I love it. Also, watch the Domains and Kingdoms of Life, and I've kind of explained the domains already. And then here's a YouTube video on how antibiotics work, which also will help you. And I've added an additional video that is just absolutely amazing. Absolutely amazing. It's an 11-day experiment that the Harvard Medical School conducted on antibiotic resistance and how fast it occurs. They made the most gigantic petri dish that I've ever seen in my life, and they incubated the outer edges of this huge petri dish with E. coli that are normally susceptible to antibiotics. And what they did was they increased the amount of antibiotics as they moved toward the middle of the petri dish. And I want you to watch how fast resistance happens. When bacteria are exposed to antibiotics, the population causes changes, causes genetic changes. They actually mutate their own DNA. And this causes changes that will allow somebody in that population to survive. And those that survive multiply and multiply and multiply and survive in that particular condition. Take a look at this video. It is amazing at how fast bacteria become resistant to antibiotics and how strong the antibiotics can be and how fast the bacteria react to that. So take a look at this video. It is just amazing. All right, your discussion board postings this week. Your initial post. Why do antibiotics target bacterial cells and not ours? First of all, an antibiotic is a chemical. It's a chemical that we take or we put on our body or however it's administered that kills bacterial cells, but not ours. That's very important, right? There are all kinds of chemicals that will kill bacterial cells, but they also might kill your cells, and we don't want that. So antibiotics target bacterial cells and not ours. So these chemicals that kill bacteria don't kill us. And why is that? Well, that's because of the difference in our cell structures. These chemicals target very specific things in bacterial cells that we don't have or that are different in our cells, like cell walls. We're animals. We don't have cell walls. Bacteria do. They have a particular substance that makes up their cell walls, and that's a huge target for 2 whole classes of antibiotics. Ribosomes. Our ribosomes are different. Our ribosomes are where proteins are made. They're little structures where proteins are made. Bacterial ribosomes are different from our ribosomes. So take a look at that. You'll find that in Chapter 20, Section 1. How our use of antibiotics increases the rate of antibiotic resistance, because bacteria become antibiotic resistant just as soon as we start using the antibiotic. That's the truth. But then, how fast does this antibiotic resistance occur? Because we overuse antibiotics and use them in the wrong ways, antibiotic resistance happens more quickly than it should have to happen. And when we do this, we produce superbugs like MRSA. I have a friend right now who just had a hip replacement. She's a lady from my church. And her wound was not healing. And they figured out that she has methicillin-resistant Staphylococcus aureus in her surgical wound site. And this is going to spell tremendous trouble for her. They're going to have to open her all the way back up, flush her out, put antibiotics in there, and she's going to have to be on stronger, different antibiotics to help treat this infection. Methicillin resistant does not mean Staph aureus is resistant just to methicillin. These bacteria are resistant to whole groups of antibiotics, and so they are very difficult to treat. A lot of people carry staph bacteria on their skin, about 25% of us. And I know this because I conducted a 17-year study on my students who volunteered for this at York Technical College, and we tested to see how many of them carried Staph aureus. About 25% of people carry Staph aureus. So it's on a lot of skin already. And then some of these bacteria become resistant to antibiotics. And that's, the example is MRSA, because that's pretty doggone common these days. Spread by contact between people, skin-to-skin contact, but also objects, any kind of objects. That's why you will see MRSA infections in athletic teams, because people share towels, rub up against the same wrestling mats, wear the same equipment, etc. This also can happen in gyms, because if you're not careful and you lay down on a bench that was touched by someone who has a MRSA infection, and you, especially if you have some kind of little scratch or open wound, you can become infected. So we're going to talk about this. Your response posts will include a discussion of whether we should be using antibiotics in livestock. Since the 1950s, farmers and ranchers discovered that if you put antibiotics in the feed of normal, healthy livestock, chickens, turkeys, etc., they grow faster, they produce bigger eggs, they produce more meat. And so this has been a common occurrence since the 1950s. Is that a good thing? That is not a good thing, because if the animals are not sick, we're exposing their bacteria unnecessarily to antibiotics, which does what? Leads to antibiotic resistance. So that's a negative. Are there positives? Yes, there are positives. I already mentioned a few. So we're going to have a discussion about that in the response post. Remember, initial post due by Thursday, 2 response posts are due by Sunday evening. Then you're going to begin your final project. Week 2, you should have taken a look at that list of final project topics, and I hope that you've already picked one. If not, you'll be doing that this week, because you need to start writing about them. Use the Milestone 1 guidelines and rubric, which are in the instructions there about how to do this. Use the correct format. This is an outline format. This is not an essay. Not an essay. You have a template for this, and you should use that. All right, here are the guidelines and rubric for Milestone 1, which is the first part of your final project. First of all, you have to choose one. Last week, I went over an introduction to all these different topics. Let me move my face over. There you go. I went over just the basics of these different topics. And if you want to use something different, that is fine, as long as it's biology related, and I approve it. If you want to do something that's not on the list, you have to email me, m.sigmon@snhu.edu, and I will either approve it, modify it, have a discussion with you about it, whatever it is. So let me know if you want to do something other than something that's on the list. Use the Shapiro Library. I mentioned that last week when we talked about resources, because it's a wonderful resource that we have at SNHU. All right, and then the critical elements. I want to touch on these down below. Notice the outline format. You are going to use this outline format. So Roman numeral 1 is the introduction, and you should have Roman numeral 1 and a word, introduction. And then under that, you are going to describe the topic that you selected. So if you chose population ecology, a description is not studying populations. Nope, it's more than that. Describe. Describe means give a lot of detail and explain to someone what this particular topic is. Why is this significant or interesting to you? You need to explain that. Why is it significant to the field of biology? The reason that we have these topics in a list is because they're hot topics in the world today. So why is this important? And give real life examples. That's important. You will be presenting this to an audience. You know, this is supposed to be as if you're presenting to an audience. And so supporting your information that you're presenting with real life examples is very beneficial to your audience for them to see why this is important. All right, the second section is the topic section. Where does this happen? Which ecosystems? Which part of the world? Which environments? Is it a specific location? Is it everywhere? Like climate change, right? Climate change affects the entire biosphere. That's every part of the earth, from the top of the atmosphere to the depths of the ocean where there's life, right? But if you're talking about, say, you want to choose population ecology of loggerhead sea turtles off the coast of South Carolina, which I'm really interested in, then where does that topic affect anything? Okay, out in the ocean, the pelagic ocean, that's out in the depths of the ocean, but also on the shoreline, the beach, because the female turtles come up and lay their eggs there. Where? Along the coast of South Carolina. So that's much more specific. Where? Where does this happen? How does it happen? Why is this a problem? How is it happening? Explain all of that. Who benefits from this? So this is probably a problem, right? So if we're talking about loggerhead sea turtles, then they are, their population just keeps diminishing. And we hope that they don't become extinct, but they surely could unless we try to protect them and conserve their population. So who's benefiting from the decline in this population? Somebody is. It's probably going to be fishermen. You don't call them fishermen anymore. What do you call them? People who fish, who use drag nets because they're snagging up the loggerhead sea turtles, and then if the turtle becomes ensnared in the net and can't breathe, then it will die, right? So people who fish can detrimentally affect the loggerhead sea turtle population. And then people who boat, who run over them and cut open their shells. You know, the list goes on. Pollution. You've seen videos of sea turtles with plastic straws in their nostrils and that kind of thing. So, okay, so you're getting the idea there. Who, where, how, who, and your introduction. Those are the 3 parts of milestone 1. My tip of the week is to put your assignment in the correct format. So the journal assignments are essays. Yeah, questions to answer, but they're essays. Milestone 1 is not an essay. It's an outline format. You're given the template. You're given the instructions that say to use the outline format, and so you need to do this. There are also more instructions there about producing this as a Word document, which is perfectly fine. You can use other applications besides Word, but, you know, Word's most common. Double spacing. Double spacing is important to me just so that I can read it. So don't do single spacing. It helps me to read it on my computer if it's double spaced. Now, do I care if it's 12 point times New Roman font? No, just make it readable. You know if it's readable or not. 1-inch margins, I don't care about the margins as long as I can read it. Just make it where I can read it. 1 to 2 pages is good, awesome. Sometimes in these assignments you'll get word counts. I don't care about that either. Just cover the material that you need to cover. That's all I ask. Provide in-text citations. If you're getting this information from somewhere else, you have to attribute it to that source. So use in-text. That means in the body of the work, citations, as well as a reference section. For all 3 of your milestones this term, you're going to be including a reference page because you will be using citations. You don't get all this information straight out of your head. You don't know statistics, right? You don't. So you have to be getting this information somewhere. So use your references. Use the Shapiro Library to find your references. All right, with that being said, I hope that you have a wonderful week number 3. Let me know if you need anything. m.sigmon@snhu.edu. Just let me know if you need anything. Have a wonderful week.